The contributions of Barbara McClintock, discovering jumping genes. Welcome back to our channel. Today, we're exploring the groundbreaking work of Barbara McClintock, a pioneering geneticist whose discovery of jumping genes transformed our understanding of genetics and the behavior of genes within organisms. Despite facing skepticism and challenges, McClintock's innovative research laid the foundation for significant advancements in molecular biology and genetics. So, who was Barbara McClintock, and what were her key contributions to science? Let's dive into her life, her discoveries, and the lasting impact of her work. Barbara McClintock was born on June 16, 1902, in Hartford, Connecticut. Growing up in a family that valued education, she showed an early interest in science and nature. Her father was a physician, and her mother was an accomplished pianist and artist. This supportive environment encouraged her curiosity and independent thinking. McClintock attended Cornell University, where she initially pursued a degree in agriculture. She quickly gravitated toward genetics and cytology, earning her Bachelor of Science degree in 1923, followed by a master's degree in 1925 and a PhD in 1927. During her time at Cornell, she conducted research on maize, corn, which would become the focus of her groundbreaking discoveries. In the late 1940s, while studying maize genetics, McClintock made a revolutionary discovery. Certain genes could move or jump within the genome. This phenomenon challenged the prevailing notion that genes were fixed entities arranged linearly on chromosomes. McClintock observed that some genes could cause chromosome breakage at specific locations. She identified a locus on maize chromosome 9 that could change its position within the chromosome. This was the beginning of her exploration into what would later be known as transposable elements or jumping genes. Through her experiments, she discovered two key elements, AC, activator, and Ds, dissociation. The AC element could enable the movement of the Ds element, which caused chromosomal breaks. Depending on where they inserted themselves within the genome, these elements could reversibly alter the expression of other genes. In 1950, McClintock published her findings in a landmark paper that summarized years of research on transposable elements. She demonstrated how these mobile genetic elements could influence traits such as kernel color in maize by turning genes on or off. Despite the significance of her discoveries, McClintock faced skepticism from the scientific community. At that time, many geneticists adhered to the belief that genes were stable and fixed in their positions on chromosomes. Her concept of mobile genes did not fit neatly into existing frameworks. McClintock described the initial reaction to her work as one of puzzlement and hostility. Many scientists found it difficult to accept that genes could move within the genome. Undeterred by this resistance, she continued to gather data and refine her ideas. Over time, as molecular biology advanced and techniques for studying DNA improved, McClintock's work gained recognition. In 1983, she was awarded the Nobel Prize in Physiology or Medicine, becoming the first woman to receive an unshared Nobel Prize for her discovery of transposable elements. Her groundbreaking research has had profound implications for genetics. Understanding genetic variation. Transposable elements play a crucial role in genetic diversity and evolution by facilitating mutations and rearrangements within genomes. Applications in biotechnology. Today, transposons are utilized in genetic engineering and biotechnology for gene therapy and crop improvement. Influence on modern genetics. McClintock's work paved the way for further research into genomic stability, gene regulation, and epigenetics, fields that continue to evolve today. Barbara McClintock's journey reflects resilience and dedication to scientific inquiry. Throughout her career, she emphasized the importance of intuition and observation in scientific research. she believed that understanding nature required patience and a willingness to explore unconventional ideas. Her commitment to pursuing knowledge despite adversity serves as an inspiration for future generations of scientists, reminding us that groundbreaking discoveries often arise from curiosity-driven exploration. In conclusion, Barbara McClintock's contributions to genetics have fundamentally transformed our understanding of how genes function within organisms. Her discovery of transposable elements not only challenged existing paradigms, but also laid essential groundwork for modern genetic research. Thank you for joining us today. If you enjoyed this video or have any thoughts, please leave a comment below. Don't forget to leave a like and subscribe for more.